Thank you very much for coming along, and it's a great pleasure to welcome Carla Finn, who I know well because she's in my research group. And it's wonderful to have her here today to talk about understanding wild population structure and developing genomic resources for Yellowtail Kingfish. Carla, over to you. Welcome.
Thanks, Pete. And yeah, thank you everyone for being here. And thank you as well to Genomics Aotearoa for giving me the opportunity to talk about this project. So as Pete mentioned, this project is about uncovering the wild population structure of Yellowtail Kingfish, as well as developing genomic resources for this species to support selective breeding, as support for the Māori-led aquaculture. So as part of this project, sorry, this isn't working for some reason. Okay, that works. Yeah, so the points that I will be covering today in relation to this project involve a brief introduction to aquaculture within New Zealand, as well as the goals of this project and the progress that we have so far. So far we have a final genome assembly, and I'll be discussing the methodology and some of the initial results that we have for that, as well as some of the initial statistics that we have on wild data sampling. Before getting into the methodology and the results, I firstly just want to thank all of the partners which have collaborated with us on this project. So this project is supporting the Smart Māori Aquaculture Initiative, which is an initiative of Aotearoa, and this is exploring pathways to sustainable aquaculture within the Bay of Plenty region. So samples for the Bay of Plenty were sourced through fishers which are local to this region, as well as help from Chris Inslee, who is also a lead on this project, a CEO of Aotearoa. And our lab group also has a close relationship with Mark Fenwick of the Aotearoa Fisheries Trust, and he advises us on the best practices when working with Tonga species, as well as sampling for the Wellington region. So we are, of course, very grateful to these people for their continued enthusiasm and help with the project. And we also have a growing list of collaborators and worthy acknowledgements who are also helping with us for this project. So yeah, very grateful to those. To begin, I want to introduce some aquaculture background for New Zealand. So aquaculture is a controlled cultivation or farming of any aquatic species, including fish. And in terms of relevance, aquaculture is the fastest growing primary sector in New Zealand. So aquaculture has many benefits. It has a low environmental impact when we compare it to terrestrial forms of farming, to many terrestrial forms of farming. And it's also highly resource efficient when compared with these alternative protein sources. So, for example, it has a higher protein and energy retention, as well as a more efficient feed conversion ratio. And currently in New Zealand, aquaculture relies primarily on the farming of 3 species. So these are the green-shell mussels, the Pacific oysters, as well as only 1 finfish species. And this is the non-native king salmon that was introduced from California in the 1890s. So there is ample opportunity to diversify the range of species which are farmed here in order to enhance food security as well as to support local communities. This is just a note to highlight the role that Genomics Aotearoa has in supporting aquaculture. So supporting initial development of genomic resources for aquaculture is important because aquaculture hasn't experienced the benefit of genomic tools yet in a similar way that other primary animal industries have. And this is particularly true for the availability of sequencing and availability of reference genomes. And the starting point for a project such as this is to understand the genetic diversity within wild stocks so that we can inform the selection of brood stocks for new species to aquaculture. So fish farming is unlike many land-based farmed species, which already have had a long history of selective breeding and domestication. With that in mind, I will introduce the Smart Māori Aquaculture Initiative. So So Aotearoa Fisheries, on behalf of Māori in the Bay of Plenty region, have developed the Smart Māori Aquaculture initiative. And this initiative recognizes that wild stock fisheries are in decline whilst the demand for protein continues to grow. So this collective is critically investigating alternative ways to supply high-value seafood for local as well as for international food markets. And it recognizes that there are no native finfish currently in aquaculture. And so they have identified Yellowtail Kingfish as an indigenous finfish with high aquaculture potential. And this is owing to its indigenous status and its high market value, both locally as well as internationally. In terms of some information on this study species, so Yellowtail Kingfish are a temperate water pelagic piscivore, and they are distributed within the Southern Hemisphere Ocean, as well as the Northern Pacific. In New Zealand, it is found from the Kermadec Islands down to the Fovos Strait. However, catches are predominantly or have traditionally been more predominant in the northern half of the North Island, but this is changing due to warming oceans. And it is characterized by its large size. So individuals are capable of reaching up to 2.5 meters and also weights of up to 70 kilograms. And because it has rapid growth as well as high quality flesh, it's especially prized in sashimi markets, which highlight its economic viability for aquaculture. At a global level, it is not a new species to aquaculture. There is currently research being done into captive Yellowtail Kingfish at various locations, including in New Zealand. So there has already been research into how to improve captive fish performance and health, as well as research into optimal environmental conditions for the best health and growth, such as temperature and pH variables, as well as how to best manage parasites within an aquaculture setting. So in terms of what we already know about Yellowtail Kingfish structure in New Zealand, this is largely based on early studies of tag and recapture studies. So this was a study in 2004, which was based on tagging and meristic counts. So meristic counts mean countable structures of fishes, such as fin spines or rays. So this study showed through these measures that there were significant differences between the northeast as well as the west coast samples, so contained within the Tasman Current on the west coast and the East Auckland Current and the East Cape Current on the east coasts, and it suggested that there were little mixing between these populations. It also suggested that the east coast might be further subdivided into northeast and Hawke's Bay stocks, and this was based on unlimited tagging study exchange. I mentioned earlier that yellowtail kingfish are traditionally found in the northern half of the North Island. However, this has been changing over the past decade. So catches within southern regions have been increasing, and it's worth investigating whether this expansion further down south represents an adaptive front. If this is the case, then this population, which is moving further down south, may be genetically unique and so could be a more vulnerable stock to overfishing. In terms of population genetic studies, so there are a number of studies looking at population genetics of yellowtail kingfish globally. However, there have been no genetic studies of this species which focus solely on local waters. Those more international studies, which do include yellowtail kingfish from New Zealand, for which there are only 5 of these types of studies, these have only sourced individuals from the northeast of the North Island. So where the coordinate data was available, that's what's been plotted here. So that means that all of the genetic diversity, which might be present further south, currently is unknown or under investigated. And the methodologies which were used in these 5 studies included examining genetic polymorphisms using microsatellite DNA and mitochondrial DNA control region markers, And because there's a lower representation of genomic information in these methods than, say, using a whole a genome-wide SNP data set, and this could raise concern about their effectiveness in genetic analysis and in delineating population structure. So yeah, so none of these studies fully utilized the genomic variation available within individuals, and this would otherwise, or could otherwise, provide valuable insights into the adaptive genetic variation which exists between populations. So understanding population structure is also important for fishery management guidelines. So currently, fish stocks in New Zealand are divided according to their supposed distribution via a quota management system. And this is a legislative framework which is designed to mitigate the risks of overexploitation through allocative measures. So within one quota management area, for example, Kin 3, total allowable catches are provided for the 98 commercial species. And this relies on data regarding the abundance and distribution of populations. However, without appropriate depth of study into a species' population dynamics, such as their genetics, what we actually find could be a more nuanced biological reality. So here, for example, in this hypothetical example, we actually have 4 discrete populations of this fish species. So these will each have their own unique characteristics and so consequently have different susceptibilities to overfishing. So without careful consideration of a species' population dynamics and genetics, we can begin to see mismatches between actual biological population structure and the fishery management guidelines. This type of scenario was reviewed in 2009. And this study looked at the Northeast Atlantic fisheries, and this revealed several instances across several species where genetic studies actually contradicted the assumptions made by fisheries management. So this could indicate potential mismanagement as well as the risk of unsustainable fishing practices. So this underscores the importance of employing a comprehensive method to bridge the gap between management strategies and what is a biological reality. And just to hammer this home a bit more, this misalignment between fishery quota management area and actual population structure has already been demonstrated in New Zealand. So the previous PhD student in this lab, in the Ritchie lab, demonstrated that this was the case in snapper. So he used genetic methods and found that there was distinct population structure between the east and the west on the North Island. And he explained this as being due to major ocean currents limiting the dispersal of snapper. And so by applying these same methods, we might be able to understand whether there is also a similar genetic difference present in kingfish populations. And so with those research gaps in mind, what I will be discussing today are 2 chapters of the thesis. So this includes the genome assembly. So we've produced a genome assembly to capture the unique genetic characteristics, the diversity, and the adaptation of the local New Zealand population. As well as this, it will also serve as a strong foundation for identifying genetic variants, which might be associated with population structure. And then we also have the population genetics work. So this will use genome-wide SNP data to assess the structure of wide-ranging samples. So we'll begin by focusing on the genome assembly. So to begin with, just a brief overview of the methods that we used. So firstly, we extracted DNA from a Yellowtail Kingfish individual whole blood using the high salt protocol. We then sent this off for sequencing, for various types of sequencing. So we sent it for Oxford nanopore reads as well as short Illumina reads. So of course, Oxford nanopore reads are longer, they are easier to assemble for algorithms, but they tend to be less accurate. And then for the short reads, they are more accurate, but they could be more difficult to assemble. So the aim for this was to use these within a hybrid genome assembly. We also have Hi-C data, which we use to scaffold to anchor our scaffolds to in order to try and identify the chromosomes for our assembly. And then we also have iso-seq data from muscle and fin clip data tissues, sorry, to help us identify the transcripts, the protein transcripts, and annotate the genome. So as I mentioned, we put our breeds through a hybrid genome assembly, and then we move on to genome annotation. So we have yet to do the genome annotation, so unfortunately I won't be able to discuss this today. We're still developing the methodology for this, but we're pretty confident that we have a final genome assembly produced. So I thought maybe some of you, being genomics people, would be quite interested in more of the details surrounding the methods. So yeah, I'll just highlight some of that now. So as I mentioned, we used Oxford nanopore reads with 117x coverage, as well as Illumina reads with 116x coverage. We then put these through the hybrid genome assembler Mazurka. From that, we have our draft assembly, and we put our draft assembly through 3 rounds of polishing using Pylon. We then purged the haplotics. We used Hi-C scaffolding using this program, yet another Hi-C scaffolder. And then this gave us our final assembly. So throughout all of these steps, we continually assessed the results of our genome to make sure that we were on the right track and that we weren't making any mistakes. The way that we did this was through a few methods. So we used a synteny analysis using MeshMap, and MeshMap is an approximate genome assembly aligner. And so we aligned our genome to a priorly published S.lalandi genome, which is of a China locality. We also assessed completeness in terms of BISCO scores, as well as looking at the general contiguity stats for our assembly, such as N50, L50, and so forth. So this is just a note to say that the following results are still being edited and are still preliminary as we work through annotating and refining our genome. So please refrain from taking any screenshots or videos of these results. Of course, if you have any questions, you can ask them afterwards, or you're always more than welcome to email me and we can discuss them afterwards as well. So onto the statistics for our genome assembly. So our scaffolded assembly was 644 million base pairs long. And we had a very high BISCO score of 98.7%. And looking at our contact map of our scaffolded assembly, we had some good news because we saw that there were 24 putative chromosomes, which is what we were expecting based on previous Sariola-Lalande genome assemblies, as well as previously published karyotypes. And then when we looked at those 24 chromosomes in more detail, the span for this genome assembly dropped slightly to 642 million base pairs. And the complete BISCO score was still pretty high at 98.6, but this is a drop by 0.1%. We found that this was due to a fragmented BISCO being lost with the otherwise removed scaffolds, but we didn't consider this an issue because it's still a very high percentage in terms of completeness. So we moved forward with these 24 chromosomes that we had identified. So in terms of how our genome assembly compares, so I don't know how many fish people are in the audience, but fish are the largest vertebrate group. So they have a huge variability when it comes to their genome sizes. So for example, we have the pufferfish, which has one of the smallest known vertebrate genomes at about 390 million base pairs. And it was found in 2021 that the marbled lungfish has a genome which is much larger, and it's estimated at 130 billion base pairs, which is the largest known of any animal. So this study was actually from 2020, so it didn't include some of those larger genomes, but it did note that genome assemblies are publicly available for fewer than 1% of fish species, so approximately 240 species. So this graph demonstrates that in terms of these available genomes, their genome length and their scaffold length. So for this, our genome falls roughly around here. So it's a pretty average size when it comes to the rest of available fish genomes. However, in terms of scaffold and 50 length in kilo base pairs, our one is actually off this chart. So obviously this paper hasn't been updated to include recent high quality genomes, but it does indicate, one, that we are perhaps done quite a good job, but I think it also means that, since then assembly algorithms have since improved. So this fragmentation that we tend to see with fish genomes, which are traditionally a bit harder for assembly algorithms to handle, this trend has since improved. So there are more contiguous genome assemblies becoming available because of these new algorithms. Yeah. And it's worth noting actually that fish genomes do tend to have quite complex genomic features. So they tend to have quite high duplicated loci, especially in salmonids, as an example. So that can often present a problem for assembly algorithms, but I get a bit more into that in a bit, I think. So, yeah. So this is just zooming in a bit onto the genus Seriola and looking at how our genome assembly compares to other publicly available genomes. So there are 9 Seriola genus genomes available. I've just picked a selection of these just to demonstrate that there is a range of qualities. So as I mentioned, some of these genomes are very fragmented. So for example, I have a very high scaffold count or a high N50 count. And we also have a range in terms of completeness. Some of them don't even have their completeness available. Some of them are extremely high. There is also this other chromosome level assembly, which is what I compared my genome to within the synteny analysis for MashMap to check that we were on the right track. So it's a chromosome assembly. So this means that the chromosomes have been identified through Hi-C, like with our assembly. But all of this to say, despite this range of qualities, our one does sit pretty high up in terms of its completeness as well as its contiguity, indicating that it will be a useful resource to use for future genomic studies for Seriola lalandi based in New Zealand. So I appreciate that this slide has a lot of information on it. Because we had a chromosome level assembly, as well as one which was quite high quality, we can begin to explore the genomic architecture of this assembly. What I think is notable within this assembly is that we have identified putative telomeric regions, and these were identified across the assembly at either end of each chromosome, suggesting that we may have a telomere to telomere assembly. And as well as an extensive representation of the genome. Something else that might be interesting is there are these peaks in the DNA transposon density. So DNA transposons are a type of mobile genetic element that may disrupt gene function, and the density could be an indicator of potential genomic variation or instability. So once we annotate the genome, we can also begin to have a look at gene density on this graph, and we can investigate whether these DNA transposons are correlated with gene density. And looking at the repetitive elements in our genome a bit closer. So repetitive elements within our genome represented 22.24% of the genome. This is similar to the priorly published S.lalandi genome. So that's great that it's nothing unexpected. We do have a large number of these repeats as unclassified. So this is something that I am rerunning to try and see if I can find a way to classify these repeats, because I think that would give us some interesting information. So watch this space for that. But looking at the percentage of repetitive elements in genomes across various fish orders. So again, variation in repetitive elements are concurrent with this variation in genome size that we have within fish. So zebrafish, for example, are known for having a very high transposable element and repetitive DNA content at 53%. As I mentioned earlier, Atlantic salmon genome has about 30 to 35% and repetitive elements comprising in their genome. So it's a big variation. Our one, again, affords about within this area. So nothing too crazy going on, and we don't want anything too crazy going on because it means that we're on the right track in terms of what we do know about Seriola. So that's good news for us. We were also able to identify, extract, and annotate the mitogenome. So this mitogenome contained all of the expected genes, RNAs, and tRNAs. It's a pretty standard mitogenome with no extensive duplications. And also it is 16,535 base pairs, which is the exact same length as the priorly published mitogenome for Yellowtail Kingfish from Japan. So this just adds further confidence to our accurate genome assembly, as well as, of course, being available for future research questions down the line. I mentioned earlier that a MASH map was used as a means of which to have a look at synteny between our assembly and the priorly published S.lalandi assembly from China. This isn't using MASH map, but it is another aligner, but instead of being an approximate aligner, we used a more accurate aligner. So we actually aligned these genomes using NUCMA. And then we calculated feature differences using DNA diff. So we used a minimum percent identity of 95% and a minimum length of 500. And what is just clear looking from this, so we have our genome assembly on the right and the China one on the left, is there are quite a lot of differences between these genome assemblies. So there is a lot of within species genome variation. With the majority of these being breakpoints. And that's quite interesting. I thought it would be worth mentioning here that there is contention over whether there are 3 different species of Seriola across the Pacific, including between China and New Zealand, or whether these are just 3 individual populations. Because of that, you will often find a variety of different synonyms used for Seriola lalandi, including Seriola aureovittata, as well as Seriola dorsalis. So there's questions over whether these are still different species or different populations. I think the most recent research is suggesting that they are different populations. But of course, having genome assemblies for individuals spanning these proposed populations can help add to this debate and can help clarify in that debate a bit more. So, yeah. So the next steps for our genome assembly would be, as I mentioned, we want to annotate gene structures using a mixture of methods. So homology-based prediction, de novo transcription, as well as using our RNA sequencing data, which is derived from muscle and fin clip data. And then based on this annotation, we'll use gene orthologs to assess relation to other members of the Seriola genus. Again, potentially contributing to that discussion about whether Seriola lalandi is 3 different species or indeed just 3 different populations. And based on that, we can also assess the unique genomic features of the New Zealand Yellowtail Kingfish individual. So that is everything that we have so far for our genome assembly. And now moving on to the population genetics. So an overview of the methods that we are using. So we will first, of course, extract DNA from wild Yellowtail Kingfish fin clips using a high salt protocol. We then send this off for low coverage Illumina sequencing, and for this we are aiming for about 10 times coverage. From that we can then identify our SNPs. So that involves aligning the Illumina reads to our high quality reference genome, calling the SNPs and then putting them through a filtering pipeline. And with that information, we can then move on to our population analysis. So assessing the degree of population admixture, as well as identifying supposedly adaptive SNPs. Again, I wanted to go into that in a bit more detail for those that are interested in the methods that we are using. So we have our Illumina reads. When we align them to the genome, we chose to use Paleomics for this. We then have our raw SNP dataset. So we then filter our SNP dataset for basic quality measures. So this is adjusted for number of alleles, for the quality score, for depth, for the missing sites, minimum allele frequency, as well as allelic imbalance. And from that, we then want to generate two different data sets. So we want to detect reproductive population structure with neutral SNPs, as well as detecting adaptive population structure using outlier SNPs. And for this, we will be using Outflank. On both of these data sets, we can then begin to have a look at the population structure, whether it's adaptive or neutral, and this will involve admixture analysis, FST analysis, and so forth. So, so far we have 52 samples for our population genetic analysis, with more expected throughout this year. So the majority of these are from the Bay of Plenty region, and that is because we are focusing on this region for Aotearoa to see if there is indeed any genetic differentiation within their area. And so just zooming in on that location a bit more, as you can see, we have a range of different sites from which we have samples collected, and that was really important for them that we sort of get a broad picture of the genetic diversity for this species in that area. So unfortunately, I don't have any data regarding the SNP filtering just yet. We're currently working on what are the best filters to apply for this data. So we're still at the population sample collection stage. So some next steps would be, of course, to secure more samples. We might have opportunity to collaborate with Australian colleagues to obtain Australian-based samples. Again, that's quite interesting considering the discussion around different populations or species across the Pacific, but it could also serve as an interesting outlier group with which to compare our New Zealand samples to. And then, we will also determine the filtering steps that we will use for our raw SNP dataset, which we already have obtained for the first 52 samples. So that is everything I have for today. I appreciate it's a bit of a shorter talk, but I just wanted to focus on the methods that we have so far, because I'm quite happy with them. And I think the genome assembly is pretty exciting. So that's everything. Yeah. And yeah, thank you all for listening. Yeah.
Thank you, Carla, for a very interesting and well-presented talk. Very informative. Thank you.